Bismillahirrahmanirrahim. In this video, the various features of the granulation tissue are discussed. The granulation tissue is the transient specialized organ of repair which replaces the provisional matrix. It is present only where and when needed, like a placenta. When required, it is present, and when not required, it is not. This granulation tissue is fluid rich. There are cells that provide immunoglobulins. There is presence of antibacterial peptide such as defensins, and various growth factors are available for the ongoing processes in this granulation tissue that we are going to discuss. Grossly, the granulation tissue is glistening and has got a pebbled surface just like this, and microscopically it is a mixture of fibroblast, mononuclear cell, RBCs, extracellular matrix, and thin-walled capillaries that are lined by single layer of endothelial cells, and these are surrounded by pericytes. The key cellular processes that are involved in the formation of this granulation tissue are recruitment of monocyte by chemokines and damaged matrix fragments. Plasma cell later become prominent in this granulation tissue. There is role of macrophage as the activated macrophage shift from pro-inflammatory M1 to alternate type of the macrophage or constructive type of macrophage that are called M2 type. They release growth factor and cytokine that is responsible for angiogenesis, activation of the fibroblast, and remodeling of the matrix. It provides blood supply to fibroblast and inflammatory cells and supply antibacterial peptides such as defensins, immunoglobulins, and growth factor to this environment. This granulation tissue is highly resistant to bacterial infection that allows safe anastomosis in non-sterile environment such as colon, where 1/3 of the content is bacteria. This set of figure is collectively demonstrating the dynamic process of the tissue repair from the macroscopic view of granulation tissue formation over here to the microscopic detail of angiogenesis and fibroblast activity to formation of mature tissue structure, scar tissue structure. All these are underpinned by cellular and molecular mechanism that are illustrated over here in the center one. So, in this gross picture, over here you can see that this is a skin wound that is covered with granulation tissue. This granulation tissue that is indicated by this arrow appear as moist red granular surface. The tissue fills the wound and provides a scaffold for further healing, including reepithelialization and remodeling. And over here in this microscopic picture, in this histologic picture, you can see that this granulation tissue is immunostained to highlight newly formed capillaries. The brown staining outline, this brown staining outline indicates the rich vascular network that is characteristic of granulation tissue. The endothelial cell proliferation is essential for supplying oxygen and nutrient for the healing process. Then a close-up histological or high power view of the granulation tissue. It shows numerous proliferating fibroblasts. These elongated spindle-shaped cells are fibroblasts with are present with newly formed thin blood vessels, and there are many macrophages or inflammatory cells interspersed. This loose matrix forms the foundation of repair tissue. Then a later stage of the connective tissue formation is shown in this picture. Here, increased deposition of collagen fiber and reduced cellularity indicates the progression toward the scar formation. Blood vessels are more mature and less numerous. In the center, the gradual process of angiogenesis is depicted. There is first vasodilation in response to VEGF and nitric oxide. Then there is degradation of the basement membrane that allows endothelial migration, and later endothelial migration occurs. And this endothelial migration toward the angiogenic stimulus. Then proliferation of the endothelial cell occur, and this endothelial cells behind the leading front, they proliferate. Finally, later these endothelial cells, they form capillary tubes, and there is recruitment of pericytes and deposition of basement membrane occurs to stabilize the new capillaries. This histological image shows a well-formed granulation tissue, an essential component in the wound healing. There are capillaries. These are the capillaries. These capillaries are well formed. You can see that numerous small thin walled capillaries. These are the capillaries that are marked by red, and these are packed with red blood cells which provide necessary oxygen and nutrients to the healing tissue. There are many interspersed fibroblasts, and these are spindle-shaped cells that are scattered throughout the tissue, producing extracellular matrix component like collagen that is essential for forming the new connective tissue scaffold. There are loose extracellular matrix. This is the light pinkish background that is showing this loose extracellular matrix. They are actually immature connective tissue matrix composed of collagen, proteoglycans, and fibronectin. Many interspersed inflammatory cells are also seen over in this picture. These are the inflammatory cells, mononuclear inflammatory cells such as macrophages and lymphocytes. These are interspersed and they play a role in clearing debris and releasing growth factor, and they orchestrate the process of tissue repair. The granulation tissue is a hallmark of early tissue repair, replacing the initial fibrin clot and provisional matrix, and it sets the stage for scar formation and tissue remodeling. This histological picture is showing wall of an abscess. Over here, this area is granulation tissue, while here, this is the purulent exudate or organizing fibrin that is present within the abscess cavity. The pink-purple cellular area, this pink-purple cellular area is mainly the granulation tissue. It contains numerous proliferating fibroblasts, newly formed capillaries, that is the result of angiogenesis, and many scattered inflammatory cells, mainly composed of macrophages and lymphocytes. The tissue is pinkish due to abundant extracellular matrix deposition in this area, and this is mainly collagen which is being let down by the fibroblast to replace the damaged area. This phase represents an active repair phase where the body is rebuilding the damaged tissue framework. Over here, this area is organizing fibrin or necrotic tissue. The loose red purple granular material in this area, present necrotic debris, fibrin, or remnants of inflammatory exudate. There is little evidence of acute inflammation as shown by numerous neutrophil and loose fibrin meshwork. This kind of histological pattern is typically seen in healing wound organizing pneumonias or infarcted areas where the body first contains the damage with fibrin and inflammation and then progressively replaces it with organized granulation tissue to restore the tissue integrity. This is a granulation tissue under high magnification under hematoxylin and evasion stain. There are fibroblasts. These are spindle-shaped cells. These are the fibroblasts that are spindle-shaped cells, and they have got elongated purple stained nuclei. Elongated purple stained nuclei, and they are scattered throughout the pink extracellular matrix. These cells are actively producing collagen and other matrix protein. There are capillaries. These are thin-walled capillaries, and they are lined by endothelial cells and filled with blood RBCs. These are bright red RBCs that are filling these capillaries, and this is indicative of active angiogenesis. Macrophages and mononuclear cells over here, many mononuclear cells are also present. Many mononuclear cells are seen in this environment. Macrophages and lymphocytes are there. Large cells with the pale cytoplasm and slightly irregular nuclei are macrophages, and they are part of the inflammatory and repair process, and they help to clear debris and orchestrate the healing. And lightly eosinophilic material in this picture, the lightly eosinophilic material that is present in the background with loosely arranged fiber is and these loosely arranged fibers are rich in ground substance, reflect the early stage of matrix deposition before dense collagen formation. So this is classic textbook microscope image of early tissue repair where granulation tissue fills the wound or damaged area, supporting the new vessels growth and preparing site for eventual scar formation and remodeling. This histologic image shows a mature scar that is stained with Mason's trichrome stain. Over here, these blue stained areas are bundles of the collagen. All these blue areas are bundles of collagen, the most prominent feature of this slide. And this Mason's trichrome specifically highlights collagen fiber, indicate dense connective tissue that is let down by the fibroblast during scar formation. Here, there is the cellularity of the area is decreased. As compared to the granulation tissue, the cellular density is much lower, and you see fewer fibroblasts and few inflammatory cells in this area that reflect the transition from active tissue repair to a mature fibrotic scar. The blood vessels, these blood vessels, these are now more mature and they are less red in color. They are less prominent than in granulation tissue. As many capillaries regress over time during scar remodeling, they leave behind mainly the extracellular matrix. There is dense organized extracellular matrix present, and the packed reorganized collagen provide tensile strength but has little elastic recoil, making scar less flexible than normal tissue. So this image in summary shows a late stage healed area where the active repair phase has ended, leaving a dense collagen-rich scar with limited vascularization and minimal cellular components. Thank you for watching this video. If you have any query, please write in the comment box.